We're going to get started again. I'm going to pass off to John to continue our COVID discussion. Of
Great.
course, feel free to put questions in the chat.
Okay, great. As always, starting with disclosures, I don't have any disclosures. Big Pharma and Pfizer are not paying my salary here. I wish they were, but they're not. So I thought we would take the next sort of 75 minutes talking about the controversies around boosters. And kind of crazily enough, when we set up the session, I never dreamed that we would literally be talking about it at the moments that these decisions would be happening. ACIP, ACIP is actually literally meeting as we speak right now and are discussing whether boosters should be used or not. And so I thought I would frame this conversation and share this conversation first with some objectives. I want to quickly review the mechanisms of actions for some of our current approved vaccinations. Just as a reminder, there was a longer session that we did on this topic of vaccinations and their mechanisms, and we went into a whole lot of detail back in March 18th, and it's on our YouTube channel if you want to watch that video. But I will share a little bit about it, just particularly for those that were not there, so we can be on the same page about some of the important ways in which they're working, and also some of the differences between various types of vaccinations, so that they're that it's clear in your head. So I'm going to list a few differences between the current mRNA vaccinations and where they're different and subtly different. I'm going to spend a lot of time talking about the regulatory and advisory process and walk through exactly what it means to be an emergency use authorization versus full authorization. How is it that the FDA works, and how is it that separate from the ACIP, and what is it that they do, and why are they two different regulatory bodies? And then really at the end, I want to spend time sharing the data that that actually the FDA reviewed. And I'm going to use in many cases the exact slides that were used in last week's meeting. And I want to actually do it as if you guys were on the FDA advisory group making the decisions and discussions about boosters, because I want you to see the same data that they had, and I'm curious about whether we as a group come to the same decision that they came to when it came to making the decisions about boosters. I do have the chat box open, but it is a little hard for me to see at the same time that we're doing this. So please feel free to unmute yourself and holler if you have a question as we go along, but we'll be covering a lot of material here. I'm actually going to start with a Zoom poll here, a survey question. And without actually going through the data yet, I want to ask sort of ahead of time how you would answer this question. This was literally the only item on the agenda, only decision item on the agenda at last week's FDA meeting. And this is the question: do the safety and effectiveness data support approval of the Pfizer Mrna booster dose administered at least 6 months after completion of the primary series for use in all individuals older than the age of 16? And so please go ahead and answer this poll. Again, just knowing what you know, you haven't reviewed the data yet. I want to see sort of where you're at ahead of time. Okay.
Giving a few more seconds for people to answer. Okay, we pretty much got a quorum there.
Okay, let's see it. Okay, yeah, so very, very close, right? 58 to 42%. Fantastic. I want to see if those numbers change as we go through this. And then let's do this. Let's see here. Oops, Joelle, go ahead and launch the, or Meg, go ahead and launch the second question. So this is really not as you as the potential advisor to the group. Based on what you understand now about this vaccine, would you personally try to get a booster dose soon?
Great.
Yeah, let's see the answer. Yeah, so yeah, do as I say, don't do as I do. Okay, so yes, I think this is interesting to keep in mind as we think through and think about these data. Okay, so let's actually go through and as just a very quick whirlwind tour of how these vaccines work. Okay, so I'm going to be talking about two kinds of basic vaccines. There are several other different formats of vaccines, but the two main ones that we have approved in the US, the first, of course, is the mRNA vaccines. And the two main ones we have here are the Pfizer BioNTech. BioNTech is the German company which actually developed the mechanism, and Pfizer was the one that worked with them and is distributing it. My friends from Germany, including my sister-in-law who's German, often point out that it should be BioNTech Pfizer instead of Pfizer BioNTech, but you know, we all have our biases. If you're curious about why it's called Comirnaty, I think that this is because it's got the RNA in it. I think that they were hoping that it would become sort of like the way we use AB at the end of monoclonal antibodies, but it doesn't seem to have taken. Moderna's version is licensed as SpikeVax, which always makes me think of spike ball and something. I'm not sure if I like the name, but it is what it is. And then we're going to very briefly mention a curiosity, which is CureVac, which I'll go over exactly what it was. But CureVac was a third candidate that had a lot of hoopla. It was, the studies were done in Germany, and it was an utter failure. And we'll talk about why that was. Okay, so let's remind ourselves about how mRNA vaccines work. And so the SARS-CoV virus, it's a single-stranded positive sense RNA virus, encapsulated RNA virus. And so what the researchers have done, the vaccine developers have done, is essentially taken the chunk of the RNA, the messenger RNA, that encodes for the spike protein. And they've taken that and encapsulated in lipid nanoparticles surrounding the mRNA. What the lipid nanoparticles containing the mRNA do then is once it's in the body, it gets absorbed into the presenting cells, and then in your cellular machinery, it gets translated into the spike protein. And the spike protein of the coronavirus is a trimer. It's a three-part protein. And each of these spike proteins combine and produce the spikes. And these are then protruded out for antigen-presenting cells and are used to help formulate and produce antibodies against those with the normal mechanism involving macrophages, helper T cells, B cells, and all the like. And you guys are all much closer to immunology than I am, but hopefully this all sounds familiar to you. Then ultimately, when the virus comes and invades, then the body is primed and ready to produce it has circulating antibodies, at least early on in the vaccine delivery. There's a lot of circulating antibodies. And then importantly, and really talked about much less, are the important roles of the B cell and T cell responses. The T cell responses in particular, where there is immunity again and cellular immunity produced, and not that well studied. There's not really good assays for it in the way that we have for circulating antibody levels. An important note here is this piece about the messenger RNA here. And I don't think I appreciated this right when we did this presentation 6 months ago in March. But one of the key questions on this is why is it that it's so hard to do this? Why is it so hard to produce messenger RNA vaccines? Why has it taken these many decades of research to produce this? Well, one of the reasons why this has happened, one of the reasons that makes it technically so difficult, is that messenger RNA, the body doesn't like messenger RNA randomly circulating through your body. You can see why it's not a good thing in terms of viruses invading your body, that there really are actually cellular and tissue mechanisms in place to help reduce random RNA from invading your own cells and producing invading proteins. And so there are enzymes in place. There's also interferon-mediated responses that actually recognize RNA that's not part of your own messenger RNA system. And one of the ways in which the researchers have gotten around this is actually to fool these enzymatic processes by actually substituting some of the uracil nucleosides, and by substituting them, it disguises it in the system. These messenger RNAs are still degraded very quickly within probably hours or several hours, or at least a half a day or less. The messenger RNA is all gone from the body. And this is one of those key messages that I share with my own patients as I talk to them. There's often this mistaken belief that it changes your genetics, right? You've probably heard this in the popular media, that it's changing your genes. The genetic material that's in there is degraded very quickly and is gone from the body within a half a day to a day at most. And so it's gone. But that's even longer than it could be if it weren't for this modification to the messenger RNA. And so one of the, I mentioned CureVac. CureVac was a vaccine candidate out of Germany. And there's a whole lot of hope about this because, among other things, they were making some changes to this to make it cheaper and easier to use. They made some decisions in their study, and one of the key differences was the dose. The dose was much smaller than the other two. And instead of using this pseudo-uracil or this modified uracil nucleoside, they actually allowed it to be quote unquote natural uracil. So just the messenger RNA fragment without any alteration. And so when they did their phase 3 trial, huge phase 3 trial, lots of excitement about it, their efficacy, vaccine efficacy against disease was 47%, much lower than Pfizer or Moderna. And so the whole effort has largely fallen apart. The other things I've highlighted here, I want you to understand about Pfizer versus Moderna, and it turns out to be kind of important, we think, is that the Pfizer dose, so the messenger RNA segment is virtually, if not exactly the same in both. They're taken from the wild type virus, the original one from the Chinese sequencing done in February of last year. So none of these are based on the variants. But the dose in which Pfizer is using is 30 micrograms for the active component versus Moderna's, which is 100 micrograms. The interval chosen in the phase 3 trials for the Pfizer study was 3 weeks, and that's the interval that we use, versus Moderna, which chose a 4-week. I'm going to come back to this pre-fusion versus post-fusion spike protein in a second, but those are the main differences. There's some minor differences in the nanoparticles, the lipid particles, but for the most part, they are exactly the same otherwise. The second mechanism to tell you about is the adenovirus vaccines, and these are the DNA vaccines. And I'm going to mostly focus on the Janssen, which is the Johnson & Johnson subsidiary, the Janssen vaccine, but also because worldwide, the Oxford AstraZeneca vaccine is probably the largest and most frequently used worldwide. Also just briefly mention those. The third one that's on the list here is the Sputnik V, or that from Gamaleya, which is the Russian laboratories. So this is also one that was produced very early, and I'll share with you a little bit about why that one has had some problems. Okay, so adenovirus vaccine. So messenger RNA-based vaccines are technically very challenging to produce. And we see that in the early days of the vaccine, when you heard a lot about how low the temperature had to be in terms of the freezer technology in some of these versions. Adenovirus vector vaccines are much more stable. And the reason for that is because you're taking the messenger RNA portion of the spike protein and actually turning it into a DNA strand, right? DNA is much more stable, can be stored at higher temperature, and it just makes sense because there's two copies, a copy and a reverse copy, and so errors are fixed much more easily, and so it tends to be much more stable. In order to be taken up into a cell, though, the adenovirus has to stimulate an immune response. So it has to be an adenovirus vector that causes an immune response to rev up so that it's taken up. But at the same time, you don't want it to have the mechanism to produce the adenovirus as an infection. So you need to have the machinery for replication deactivated for the original adenovirus. And so the steps actually, for the most part, once it's in the cell, are largely the same, except that it's, instead of the single-stranded RNA, it's the double-stranded DNA. But once it's in the cell, then it does its thing, and you produce the same 3 trimer, the spike proteins, and the same steps afterwards happen. The key features and the key differences between the two, Johnson & Johnson, the Janssen vaccine, is uses a replication incompetent adenovirus strain called adenovirus 26. And Ad26 vectors have been used previously in studies looking at Ebola virus vaccine candidates, and other adenovirus vectors have been tested for studies in HIV studies and Zika virus vaccines trials. The Oxford group, in their AstraZeneca partnership, they use, instead of a human adenovirus, they use a chimpanzee adenovirus. And so it generates an immune response, but because it's not, it doesn't have the ability to invade human cells, that's the way in which that's deactivated. The Johnson & Johnson and Oxford AstraZeneca vaccines are very successful. I'm going to contrast that. So the Sputnik V one, the one that's used in Russia widely, and Russia was exporting it to a lot of countries. There's a lot of issues with how they've done it. They've chosen a very different mechanism, which is to use a replication incompetent adenovirus 26. And then for their second dose, they use a different adenovirus vector, which is adenovirus 5. And their theory behind it was that if one kind of adenovirus was good at stimulating the immune response, that a second adenovirus would be even better. And so that was why they chose that. The reason I've highlighted a couple things, this post-fusion spike protein is one difference between the Sputnik one and the Johnson & Johnson one, but I've also added an asterisk. And it turns out that in Brazil, which was trying to buy the Sputnik V vaccine, they actually discovered that, in fact, they weren't using completely replication incompetent adenoviruses, that a bunch of the adenovirus was actually producing adenovirus. And so it wasn't a purely replication incompetent. And so they rejected this vaccine, even though they very much needed vaccines in Brazil, because you were essentially infecting people with adenovirus, which again, for many people may not be a huge deal, but for the immune incompetent or the immune people with immunity issues, it was a huge problem that we were infecting people with adenovirus infections. So the other big difference between Johnson & Johnson and Oxford AstraZeneca is this thing about the single dose, right? So Johnson & Johnson is a single dose vaccine. And the AstraZeneca one uses an 8 to 12 week follow-up. And we'll talk a little bit about the length of time of follow-up and why that's important. But this, the other big difference is that Johnson & Johnson uses a pre-fusion spike protein versus the AstraZeneca, which uses a post-fusion spike protein. So why is this important? So it turns out that the SARS-CoV virus, like several other viruses, particularly in the respiratory tract, that have these spikes, what they have is the spike is a certain shape. It binds to, in this case, the ACE inhibitors in the human cells. And then once it binds, it pops, it springs into a different form. And that different form actually is what allows it to fuse into the cell. So on the left-hand side, you see the pre-fusion spike shape based on modeling. And the right side here is the post-fusion spike. And so many of the vaccine candidates actually use the post-fusion spike formation because it's more stable and easier to work with. The Mrna, the Pfizer, Johnson & Johnson, and then one that we hardly hear about these days, which was Novavax, all use the pre-fusion spike formation. Why that might be important is that if you think about this virus that's circulating in their body looking for cells to attach to, the antibodies that are formed against the pre-fusion version might, at least theoretically, allow you to catch more of the virus particles before they fuse into the cells, at least theoretically. And so whether or not that's a huge importance, it's still not entirely clear. But if you look at some of the efficacy rates of some of the vaccines and why some of these seem to be higher in some groups than others, there is a general pattern that the pre-fusion spike protein antibodies seem to be better at protection than the post-fusion ones. All right. I'm not going to talk about the rest. Novavax I touched on, which was hugely promising, huge production problems. Again, very, very hard technically to produce these, but this was meant to be the next best thing for those that really didn't want vaccines that were based on genetic material, that this was a protein-based vaccine. And then the ones at the bottom, they're inactivated virus vaccines, which are really our oldest form of vaccines, are still being used widely in many other parts of the world, but in general, less durable effects and less robust effects seen with those. I'm going to pause there and actually, because I'm going to move on and change topics a little bit. But I know I covered a lot of stuff about vaccines. If people had questions about how the vaccines work, I'm happy to take them now. I'm not seeing anything in the chat yet, but okay. All right, if anything occurs to you, don't hesitate to put it in the chat. Since I gave this talk back in March, a bunch of things have happened, and many of these we're all familiar with, but I'm just going to list them here because it's amazing to think how 6 months we've seen so much when it comes to these vaccines. First of all, like back in March, we were starting to see this event, this vaccine-induced thrombotic thrombocytopedia, which I listed as HIT-like because in fact that there is some similarities and overlap with the mechanism. There seems to be an anti-platelet 4, platelet factor 4 kind of antibody thing that's happening in some of these. This really does seem, in fact, limited to the adenovirus DNA vaccines, so the Johnson & Johnson and the AstraZeneca one, and it was particularly prevalent in young women. When I say prevalent, it's not very prevalent at all, but there was definitely a predominance of young women compared to men or older patients. And then the other thing, of course, that we saw and we continue to have an eye towards was these episodes of myocarditis and pericarditis. And there is definitely a clear predominance in young men and boys. And this caused concern as we were talking about lowering the ages in which we were giving these. And in fact, there was worry, were we sacrificing and seeing more problems as a result of the myocarditis that would be life-threatening in people who at least seem to be less at risk for infection? VA folks, yes, we'll post those. What we've seen since then in terms of the adverse events monitoring seems to be that, yes, in fact, that there are more events of myocarditis, but that the events actually are much, much milder, certainly much milder than the myocarditis that's occurring as a result of SARS-CoV infection, but also much milder compared to other viral myocarditis episodes. The pattern seems to be people who have very mild chest pain or chest discomfort. Findings are seen on EKG and or cardiac imaging. And they're often hospitalized for a day or two just to make sure that they're doing better, and then are released usually within 24, 48 hours from most children's hospitals. And so although it's seen, we have seen some more severe cases of it, but the pattern seems to be less intense than some of the bad cases of myocarditis we see in other situations. Then the other thing that the other note here is that we're very fortunate. We live in a very resource-rich country, obviously. And we had the option of following the vaccine dosing schedules as originally planned. And so we opted for the Pfizer vaccine, we opted for a 3-week interval because that was what the phase 3 studies showed, and for the Moderna, a 4-week interval. In other countries, and I've highlighted here the UK, where their vaccines weren't as quickly available in some of these other countries. UK is still a resource-rich nation for sure, but but they had shortages. And so many other countries opted for a strategy where they gave first dose to as many people as they could. And then they followed up with their second dose when the vaccine supply caught up. And so what they found in the UK was that actually circulating antibody levels were about 3.5 times higher in those where they waited 12 weeks for the second shot rather than those who waited only 3 weeks. So immunologists can explain this better than I can, but there is really good, actual plausible rationale why that might be, that doing so might actually prompt a higher response rate if you have a longer interval between the two doses. And that's going to come up in our conversations about boosters. Of course, we saw the extension of the emergency use authorization for 12 to 15-year-old children. And this is the part that has caught a lot of attention, which is that we do see, in fact, that there's decreasing circulating antibodies and increased infection among immunocompromised. The immunocompromised have had third doses approved for Pfizer and Moderna in these individuals, and that was in August. And then at late August, we saw the full FDA authorization for Pfizer, and the submission is currently under review for Mrna. Okay, so those are events since the last 6 months. I want to make sure that we're on the same page about the regulatory approval, and I have some of your faces up. Just by show of hands, is anyone else confused, or is it just me by why it seems that we have such a redundant process involving two different groups, like with the CDC and FDA? Was anyone else confused by this? Yeah. Yeah, it seems like a little odd, right? Yeah, thank you for admitting your confusion. Why is it that we have two separate processes? Well, let's talk through sort of, I'm going to share with you a couple of slides. These are from CDC and a couple of other places. And so I'm going to repeat some of the information here, but they're graphically depicted in a couple of different ways, just because I didn't like any of them completely. But let me walk you through. So this is from the CDC's own website about vaccine safety. And what this is highlighting here is what the normal review process looks like. What is the normal process by, thank you, Matt. I'm confused about many things, me too. What is the normal regulatory process around vaccines? And so the process is really designed more so for the viewpoint of safety, right? The view of this whole process is geared towards what is safe and what is okay in terms of the risk for a vaccine. And so what they've depicted here on the left-hand side here is that in these early phases, so like in the basic research, the discovery, preclinical studies, this is all, right? Like this is all what the scientists are doing. Then what they submit is an application for something called an investigational new drug application. And this is what's done by the drug companies and the scientists ahead of actual even phase 1 studies. And so the phase 1 studies is really geared towards safety, right? Like you want to figure out, like, if I give this, people aren't going to anaphylaxis and die right away, or this may also involve things like these studies are oftentimes in studies of like 15 people, right? It's just a small like test to see that people are going to tolerate the next phases of the studies. Phase 2 studies are about effectiveness. And so these are oftentimes larger in the order, sometimes in terms of the vaccine studies, of like several dozen, maybe 30 or 50 individuals, to look to see whether there's a signal towards an effectiveness of the vaccine, or for that matter, whatever the drug you're looking at. And then it's the large phase 3 studies, large phase 3 trials, because these are usually randomized controlled trials where you're looking at both safety and effectiveness. And for the most part, for full authorization, the general rule is that you need a large, well-conducted, well-controlled randomized trial in order for this to get reviewed. Even better is if you have 2 of them. Okay, so the normal process then at that point, once you have the phase 3 trial data, is you submit something called a BLA, which is this biologics license application. And that gets submitted to the FDA. The Food and Drug Administration is the one responsible for reviewing drugs and new technologies, and this counts as a new technology and drug. And this biologic license application is the first step that that has to be done. The FDA then, because and there's a lot of history behind why this is. If you're curious about it, Google the history of FDA and thalidomide, the anti-emetic drug that was used and caused birth defects. There's a lot of reasons why they do this, but they actually demand all the raw data. They don't accept data that's prepackaged from the drug manufacturer. They say that they want to look at, and their own scientists want to look at the material themselves, run the numbers themselves, and double check the data. And when they do so, once they've done that review, they package that up and give it to a group called the Vaccines and Related Biologic Products Advisory Committee. And so the VRBPAC, VRBPAC, this Vaccine-Related Biologic Products Advisory Committee, is a group of independent people. They're not FDA scientists. This is a group of academics that volunteer for this, and they meet usually quarterly, and they review all of these applications. And when they review this, they look at the raw data. They look at the way the new data look like. They look at the submission from the drug company, and they make a vote. They take a vote. And based on that vote, they recommend thumbs up or thumbs down to the FDA whether to approve it or not. The FDA then takes that information and makes a choice based on the recommendations of the advisory group, and then decides to approve it or not. Almost always, the FDA advisory group has a huge influence on the FDA's decision. This probably about 90–95% of the time, they just take that recommendation and absorb it. They don't always. And where this came up recently, if you've followed the news, there was recently a new medication that was approved by the FDA, where the FDA advisory group pretty much overwhelmingly rejected it. It was a drug for Alzheimer's disease, and it was one of the very first new drugs for Alzheimer's that had come out in a long time. And the FDA approval was based on a subgroup analysis that was done post hoc after essentially the study was closed, and they approved it. And so they sometimes will go against the advisory group. Then the FDA approving it is essentially opens the door for people to prescribe it. But that still does not cover yet the actual recommendations of how to deliver it. And for that, the FDA then sends it to the ACIP. So the ACIP is a CDC Review Committee. The CDC Review Committee is made up also of academics and community members and advisors. And that group meets and makes recommendations about how vaccines should be given. So why is it that you have this FDA approval of the drug and the ACIP review? The CDC typically needs to approve these for a couple of reasons. One is that they're required to approve it in order for insurance companies to cover the vaccines. Secondly, for the most part, for many vaccines that are given to kids, they control a program called the Vaccines for Children's programs. And because so much of this goes through Vaccines for Children programs, they are the ones that adjudicate whether to pay for the vaccines or not in uninsured kids, and so have a huge influence on this. And most importantly, the CDC actually handles the logistics of how the vaccines are delivered to hospitals and to clinics. And so because they're handling the logistics of the vaccines to those locations, they have to sign off on this. So in general, the FDA approves a drug or a vaccine once, and then the ACIP reviews it actually frequently. And so you may have wondered in your own primary care clinic, why is it that it seems like every year or a couple of years, the recommendations about pneumonia vaccines seem to always change? I can't keep it straight in my own head. It's because the ACIP reviews it every year, and they're taking into account new safety data, new trials, and they're tweaking this constantly. So FDA usually approves it once, ACIP review it on a rolling basis. And take into account all this post-monitoring, approval monitoring, and research stuff that happens. Okay, so that's the normal flow of how things go. This table here essentially says the exact same thing, but if I like this because it has a few other details in here. So again, vaccine development and testing, submission to the FDA for the biologics license application, the VRB PAC, the Vaccine Related Biologic Products Advisory Committee, makes a recommendation, and then the FDA decides to license. Once that is licensed, the ACIP advises the CDC on how the drug should be given, the vaccine should be given. And then this is the annual vaccine schedule that you get, and it's published in the MMWR. And then insurance companies generally will take that up and finance it, and et cetera, et cetera. All right, so all of that is the normal flow. So what is this emergency use authorization, and how did this mess things up and create both benefits and problems for us? So the emergency use authorization, it may feel like it's been around forever, but actually it was developed really after 9/11. And in 9/11, many of you may not remember this, or maybe maybe you recall this. If you remember, there was a scare in the couple of years after 9/11 where there was a series of anthrax attacks through the mail. There were people getting mail where anthrax powder was in the mail. And so in response and the concern about bioterrorism emergencies, the Congress passed a law in 2004, essentially broadening the FDA's authority to rapidly mobilize resources really geared towards bioterrorism. And so in order for this to be used, there had to be an emergency declared, essentially through either Department of Homeland Security, Department of Defense, or Health and Human Services. Plus, the Secretary of Health and Human Services had to declare that there was an emergency. And the three prongs for emergency use authorization are that one is that the totality of scientific evidence, given all the totality of scientific evidence, it's reasonable to believe that a given product may be effective. I'm going to contrast that in a second to what is required for full authorization. The second is that the known and potential benefits outweigh the material threat posed by the emergency. So, and in other words, that you couldn't wait because the danger was so great. And thirdly, and importantly, that there was no formally approved alternatives that were useful against that emergency. The contrast here is regular authorization, which requires that you have to have proof of safety, and you have to have, rather than reasonable to believe evidence, you actually had to have substantial evidence of efficacy. And usually that involves at least one big RCT, if not two RCTs. So this was originally intended for use in bioterrorism attacks. It's never been used really for bioterrorism attacks. It's been used though to use in emergency situations such as the H1N1 flu worry that happened. And what it was used for was to rapidly ramp up the production of oseltamivir, so Tamiflu. Tamiflu was very, very hard to get at that time. And so because of this emergency use authorization, it was quickly ramped up and allowed for rapid production of Tamiflu. And then it was later used for MERS and Ebola and Zika. And so I said this already, but again, this seems like a subtle difference, and it is subtle, but it is important as we talk about the boosters. So reasonable to believe that a given product may be effective may include observational studies, may include animal studies, may include smaller studies than you would say meets the bar for substantial evidence. Okay, and so we'll again come back to this when we're thinking about whether boosters should be approved. Okay, final diagram. I know I'm repeating myself multiple times, but I think it's worth repeating here. This is the timeline in which most of this regulatory stuff happens. So usually after the clinical trials have been submitted, it's submitted for full FDA approval. Usually it's 6 months. It's oftentimes in the past for vaccines, it's oftentimes 12 months between the time when it's submitted for full FDA approval and the VRB PAC meets. And then so again, this advisory committee meets, usually the FDA approves shortly afterwards within a couple of weeks. And then several weeks, oftentimes it's several months later, if not the next year, the ACIP meets, and then CDC decides, and then it goes to the public. So in the normal process, we're oftentimes talking about usually 9 to 18 to even 24 months from start to finish is the normal regulatory process for vaccines. With emergency use authorization, this is usually shortened, right? So this is as short as 2 months to go, and then it goes to the FDA approves. Here's the problem, and here's the issue with emergency use authorization. In the middle of a pandemic, all of this is condensed, right? In the middle of a pandemic, you don't want this to be many weeks later. So that's why we see this thing, which seems very odd to many people, why it is that the FDA approves, and then the next day the ACIP approves, the ACIP meets, or, in this case, it's 4 or 5 days later that the ACIP meets. Why is it that they have to happen in this way? It seems cumbersome and odd when the two things seem to be doing much of the same review. It's because the same regulatory processes are there. They still have to go through the hoops. They just push them all together. And in case of the ACIP, they're actually literally happening at the same time the FDA is making its decision. And then the hope is, and what happened with the Pfizer vaccine, right, is that with additional follow-up, then they apply for full FDA approval. Okay. All right. So a little recent history here, just current events or near current events. So August 15th, or sorry, August 14th, I believe the date is, I'd have to double check my dates. The FDA approved, the VRB PAC, and then later the FDA approved, as well as ACIP then approved, use of third dose vaccinations for the immune compromised. So that was not at all controversial. There was really good evidence to say that in these groups that a third dose was necessary to really bring up the immunity, and that 3 doses were necessary in those groups. The very next day to this news, August 16th, here's this New York Times piece. So in essence, what was happening between August 15th and 16th was that the administration was leaking the plan or putative plan to have boosters available for most Americans. In this case, they said 8 months. They later shortened to 6 months after vaccination. And they floated the date of September 20th as the date in which this would all become available. So August 16th, they're saying a date about 5 or 6 weeks after this, that everyone would be eligible for a booster. Just a few days later, you can see here, just a few days later, oops, sorry. The FDA finally approved the full approval for the Pfizer vaccine, right? So Pfizer was doing all of this at the same time. They approved and said, yes, you have our full approval for this. And if you remember, there's many good reasons to do so, not the least of which was that a lot of employer mandates really required the full force of a full approval rather than emergency use authorization. Okay, so August 23rd. August 25th, so it was actually August 24th when they submitted. So the day after they got full approval, they then sought approval for the COVID vaccine booster. So if you think back, the administration had already said that boosters would be available even before the drug company had submitted an application asking for the booster. So I showed you and spent a lot of time talking about the normal course of regulatory action. The first step in all of this is the drug company has to submit the request. So the administration was saying that we want this before the Pfizer had even submitted the request. August 31st here, during the week before this, several top vaccine experts at the FDA resigned. They didn't speak directly to the press, but there were pretty widespread leaks saying that they were very unhappy in which the order of events had happened, that the administration had done an end run around the normal regulatory process. And so these very senior vaccine officials stepped down in essence in a kind of protest against what had happened with that. And then we can't talk about vaccines without talking about global equity, right? And so the WHO, in the weeks since this announcement has been made, has been very, very strong in its language in saying that COVID boosters should not be given until the end of the year at least, or at least until, ideally until more of the rest of the world is vaccinated. And it gets complicated. The original licensing and the original agreements by the Trump administration with Pfizer and Moderna and others stipulated that the US could not repackage its vaccines that were allocated and bought to other countries. And so there was a lot of complicated regulatory stuff happening in the back end. We did find a way to be able to donate doses, but it turned out that like it wasn't as easy as just taking our unused doses and giving them out. But it's complicated, but at the same time, we pay a lot of money for the vaccines. A lot of countries can't afford the vaccines at the prices at which we pay for it. And so even though the drug companies claim that there are other reasons why they can still do this globally, in addition to provide boosters in the US, I think that there's reasons to be critical about and look critically at those claims. All right, I'm going to stop there for a moment, and we're going to actually get into the data, but ask if there's additional questions. I'm not seeing anything yet. Okay. All right. So again, I want you all to put yourselves in the role of the Vaccines and Related Biologics Products Advisory Committee. So you are now drafted into this group. You are part of this advisory committee. And I'm going to show you the exact same slides for the most part that they saw last week. I've chosen that, I should say the meeting was 8 hours long. So I'm not showing you everything clearly, but I'm choosing to show you, I think, what are the high level, high yield images and figures that they used in their discussion. And I'm not going to share with you their discussion points, but I actually want to open it up for conversation as if we're running the meeting. So I want you to stop me and ask questions about the data that you're seeing. I want you to talk amongst yourselves. I want to pause and absorb the information, because at the end of this, I'm going to come back to the question. So this was the single item on their agenda. This is a single question on their agenda, was, do the safety and effectiveness data support approval of the Pfizer booster dose administered at least 6 months after completion of the primary series for use in individuals 16 years of age or older? All right. I will preface it by saying that the FDA was very, very, shall we say, earnest and eager to see this approved. And they said up front, this was a statement from the chair of the FDA chair, as we proceed, I would ask that we do our best to focus our deliberations on the science related to the application under consideration today, and not on operational issues related to the booster campaign or on issues related to global vaccine equity. If we stray into these latter topics, the chair and I will gently bring us back into the scope of this advisory committee meeting. I'll tell you right now, I said that I wasn't going to tell you what they talked about. They did not stick with this, that they did stray. Okay, so let's get into it. Okay, so in these meetings, the manufacturer has a chance to show and share their own data. They start and they get to share the data that they have. And so I'm going to share with you the Pfizer data. They didn't share this graph, but this graph actually, I think it's a useful reminder. This is the New England Journal of Medicine article from December. And literally, this is the best efficacy data I have ever seen, ever. right, on a graph, right? Like this is the placebo group, this is the Pfizer vaccine group, and what we saw after about day 12 is that there's this dramatic split, and you see a virtually flat infection rate, incidence rate in the vaccinated group, and you see a linear straight line rise in the placebo group. Never seen anything like that. I don't think I'm ever going to see anything in the rest of my career that looks as good as this. But I will say, just to point out that, again, this is not the Pfizer presentation yet, but it's important to point out that the graph here is of incidence and occurrence, not of other metrics like hospitalizations or deaths. Okay, so those things lag. It's harder to prove those and takes longer to prove those, but this was of incidence. All right. So in their presentation, I'm going to let you read this and absorb it for a second here. This last bullet point here is the one that they highlighted. Okay. So an analysis of efficacy of up to 6 months after dose 2 shows that the initial vaccine efficacy slightly wanes over time in the pre-Delta period. All right. So they were saying the 95 or 96% was what they first showed. Then it dropped to 90% from months 2 to 4 and to 84% for months greater than 4 up to 6 months after dose 2. And they're saying that this is even before Delta that they were seeing this. They highlighted the left side of this graph, which is that, again, very, very busy graph. In all age groups, so the different lines here are different age groups. Okay, so the very young all the way up to the very old. The black line here is all the age groups combined. What they saw was that depending on how many months after vaccination, you saw that the vaccine efficacy against infections was dropping, so that by the time you get to 5 or 6 months after vaccination, you're looking at an efficacy rate against infection of only 47%. All right. So if you think about, and I'll ask you when you got your vaccine, think about that. You are all in this group that would be at the lowest, at this bottom of this, so potentially even lower vaccine efficacy and protection against infection. They did admit, they did say here that it still remains effective against hospitalizations. Okay, so the numbers drop slightly, right? But not that much. I mean, it's pretty much 87 to 88% even after 5 months or more in terms of protection against hospitalizations. Vaccine effectiveness wanes over time regardless of the variant. So the argument here is that it is not just a Delta effect. They are arguing that whether you limit your analysis to the Delta variant or other variants, that effectiveness against infection both show a degradation. The two are statistically the same. They're not statistically different between the two, but they both drop from 97 and 93% immediately after vaccination down to 67 and 53% 4 more months after vaccination. Okay. How are you raising your hand, or is that just an old raised hand?
It's an old raised hand. Sorry.
That's okay. I was excited that you were going to ask a question. All right. Again, a very busy slide, but let me walk you through what they're showing you here. What they are doing here is they're saying that if you give boosters, that the neutralization assay, so they do serum assays, looking at how much circulating antibodies there are, and they do neutralization assays, and they show that, like, if you look at after dose 2, this is on the left on each case, okay, after dose 2, and then after dose 3, so the planned booster injection, they show that your rate of titers of neutralization based on the antibodies goes up significantly after your 3rd dose booster. Okay, so again, this is a neutralization assay where they're showing this on a phase 1 kind of study. They then, in a relatively decent size study, they're reporting on side effects after 3rd doses given. And so this was, they compare it to larger groups of where they only got 2 doses, but compared to for a smaller subset that got a 3rd dose, they generally found that systemic side effects were fairly similar across the 2. So it wasn't that there's a hugely higher number of systemic side effects after the 3rd dose compared to the 2nd dose. So in the same range. I'm going to pause there and ask if there are questions about the Pfizer data, or not just questions, but any thoughts and reactions to it. Anyone convinced so far based on what they're seeing?
I guess just to clarify, it seemed like with waning immunity, even if only 40% of people at this time out still have kind of sustained immunity, they still, if they got COVID, were not being hospitalized typically.
That, I think that is a fair statement, at least based on the Pfizer data that they're showing.
Okay. Because I think then it brings up the thought in my mind of part of the reason they did not approve of booster is because if we're not preventing hospitalizations, if people are being able to get sick and be okay, then that's partly a reason not to merit a booster. But that people not being hospitalized but walking around sick are still very dangerous to those people who are immunosuppressed or older, and that is how much do we owe to those people to sustain an immunologic response, even if people who are immunocompetent are not going to be hospitalized. But
yeah,
that was my thought.
I want to interject a concept here, which is for the people who are in this field, they're trying to be very cautious about the term booster and 3rd dose. And I've used them interchangeably in this presentation, not to make a huge deal of it. For the immunocompromised, we are thinking of this as a 3rd dose and not a booster. And I think it's very confusing to the general public because someone can walk up to a Rite Aid or a CVS pharmacy and get a 3rd dose just by saying that they're immune compromised, and there's not necessarily a proof that they have to show. So we know that, in fact, that many people within the community are using this as a way to get a booster. But the 3rd dose in that case is really felt like it's necessary in order to bring people up closer to the level that the 2 doses get for in non-immunocompromised people. But yes, and again, Annie, I'm going to invite you not to worry too much about the thought process. I don't want you to try to guess the thought process of the committee. I'll tell you what the thought process was at the end. But I'm glad that you're thinking this way because, in fact, there's important ideas that you just said, which is infection rates. Protection against infections indeed is going down, and that yet the hospitalization protection seems to be holding. There's some questions here. Let me see if I can. How well do neutralizing antibody levels correlate to prevention of symptomatic disease? Rini, I don't think we have, there's biologic plausibility and we think that they do correlate, but it is not clear that if there's a threshold level or if there's a, yeah. The other part to say out loud is that it's actually, we do expect antibody levels to decrease. That's normal, right? Like when you get a vaccine, any vaccine, your measured antibody titers drop over time, but you have still the memory cells ready to ramp up and activate. And again, you guys are much closer to immunology than I am. That may also help explain why it is that the hospitalization rate may stay the same in terms of good protection versus the infection, because we think that with Delta in particular, that the early infections, that people are still getting early infections from the Delta variant, but not necessarily getting as frequently symptomatically and systematically sick. Are these people contagious though? Matt, that is a really important question. And outside of what data were presented at the meeting, there are data that suggests that indeed people are contagious during those first few days. So Matt, I'm assuming you're asking about people who are vaccinated and have quote unquote breakthrough infections. So for those people, we think that they are infectious. At least some of these early data suggest that they are, but not as long as the people who are not vaccinated. Andrew asked, let's see, does third dose produce only temporary benefit against infection, or is there a reason expected to also? We don't know, Andrew. And so this question was asked of yesterday's ACIP meeting. They asked the Pfizer executives this very question of whether it's, in fact, it was Jeff Dushin, who many of you know is the King County Public Health person and also on faculty, asked this very question of the Pfizer executives: is it going to be dose 3 and then dose 4 and then dose 5? Are we looking at that kind of situation? And what the Pfizer executive answered was they didn't know. They said they think it's only going to be 3, but they had no idea. I worry that even if hospitalization rates are lower, greater numbers of infection will perpetuate mutation and eventually. Oh, Rita, can you expand on this? Because I think this was actually an important thought process for many people who are debating this. Can you expand on what you're thinking about this?
Oh yeah, I mean, I might be thinking of this too simplistically, but essentially what I was thinking is that, for now, we know that there's greater rates of infection, right, this many months out. And so the reason that we're seeing mutations is because people are getting, the way that mutations form is essentially by people getting infections and having a mutation occur. So even if right now those people aren't getting, there's still a low rate of hospitalizations, there's more chance that variants could form that the vaccines are not as effective against, which could then change the whole picture. Is kind of what I was thinking.
Yeah, so is your thought process here that, so you would be in favor, if you follow that argument out, you'd be in favor of offering boosters as a way to reduce sort of like the circulating burden of how much infection there is in the community? Is that, did I?
Yeah, essentially just like the flu vaccine kind of situation.
Yeah. Great thought, great question. Like many things we don't know, but that is an argument that is used. And I'm going to show you in a moment the Israeli data that was used and discussed and debated, because this was part of their argument. In my mind, the main open controversial question is, do vaccinated adults younger than 65 with third dose or focus more on getting global unvaxxed their first and second doses? That is straying from the scientific questions. Yeah, so you would have gotten slapped down by the chair of the meeting. But let me actually provide you a little bit of background on this question. Is providing a booster better, or is focusing on getting a first and second dose in people better? The math gets a little bit wonky and complicated, but it isn't actually that hard for people to prove that your bang for the buck in terms of reducing community circulation is much greater if you move the bar in first and second doses. So if you increase your first and second doses in a community from, like, say 55% to 60%, you get actually more benefit from that in modeling studies than you would from giving 50% of people a third dose booster, unless rates are super high in the community, which they are, and a couple of other conditions. But in general, you're better off focusing on first and second doses. But it kind of begs the question of whether we have reached sort of that asymptotic imaginary point where we're just not going to get any more first and second doses in certain communities and certain individuals or not. So it's a question. There's more than just severe illness to consider: transmission rates, mutation, days of missed work in school due to infection. Thank you for that. That is super important. Yes, it is not just hospitalizations. There are actually, and I'll say it out loud, this was some of the questions that came up in the discussion. We don't have data on things like long COVID syndrome. We don't have data on things like in terms of infection rates in kids, like sort of the kind of cocoon immunity. We see data out of other countries where it seems like it's important, but we don't have data on whether the third doses add to that cocooning and reduction of ring immunity kind of idea. Okay, great questions, great debates. You guys could be on this totally on the scientific community commission, and you would be asked, they literally asked some of these same questions and debated the same things. And you'll be comforted by knowing that no one else had any answers either, that the same questions that you were asking and the uncertainty are exactly the same things that they were debating. So well done. Hang on. Okay, so the final point that the Pfizer group brought up was that in fact in Israel actually employed a booster strategy, and so the Israeli Ministry of Health was invited, and I'll share their data with you. But they summarized, the Pfizer folks summarized, they said that they essentially have instituted a program covering the entire vaccine eligible population 5 or more months after the second dose. And so 2.7 million people have received a third dose. This is an important caveat here, and important to know as you interpret these data. They did it in a staged way where they said, we're going to focus on the older people first, and they are doing it in a way they're starting with the oldest and moving down. So the majority of the data, and the majority of the data that I'm going to show you, is in older patients. Okay, they point out that to date there's really no huge safety concerns that have been raised in terms of adverse events compared in the third dose compared to first and second dose. Okay, so then the Israeli Ministry of Health presented their data, and they presented their booster data. So they first shared the rationale to why they did this. And so what they saw here, regardless of age group, they said when you got the vaccine mattered to how much the infection rate was. So complicated graphs. Let me focus here on the 60 and older here. So if you were in the group that got vaccinated earliest, so in January, your vaccine your infection rate was highest. So this was about 3.3 per 1,000. Versus if you got your vaccination in just 2 or 3 months, it was sort of like 1 to 1.5 per 1,000. So the time since your vaccination seems to matter in terms of your infection rate, and that this was independent of age group. This was one of the criticism that's been raised, and this was their attempt to address that criticism, because the numbers of people who were in this January group was largely those that were 60 and older. And in fact, you can see here that none of the January group is represented in these younger age groups. So in other words, whether or not there's some confounding there going on, not clear. Based on this and some other data, which I won't show you here, they've started a booster campaign, and they started this in July 30th, so end of July. And so among the 60 and older, they have given about 1 million doses. and they have started giving these, and you can see the waves in which they're starting to institute these. So the majority of their data are on the 60 and older out of the 2.8, 2.7 million doses they've given. Yeah, this is sort of an example of how not to do a graph in a presentation, because it's super complicated. But let me try to walk you through this. What they're showing here is after the bottom number here is days after your third dose. So they're arguing with some biologic plausibility that the first few days after your third dose, you don't actually expect to see much benefit from that because it hasn't had time to kick in. But they've drawn a box around after day 12, and they're saying, they're guessing, and again, that's probably some plausibility, that around that time is when the additional immunity kicks in from the third dose. And what these bars represent, it's a number-fold reduction in rate compared to the 2 doses. So it's kind of a double negative here. So what they're saying here, the higher the bar, the more protection, the more reduction in infection rate that you see. And so they're arguing that because, if this were independent of this third dose, you would see this across the board here, but really you see this curve here that's developing. And they argue that that is demonstration that it is effective at protecting against infection. And they give the number here of 11-fold greater protection from the third dose. Okay, it's super complicated. And it's a very, very busy graph. I'm going to take some of these questions a little bit later, or comments a little bit later. They also argue that this actually has, and this is to Arit's point about the community protective effect. So if you remember, the reproductive number, the R0, is the reproductive number. The R effective is the observed reproductive number. So the R0 is the theoretical reproductive number, and actually what's happening in the community is represented by this effective reproductive number. And so they are drawing lines here of when the boosters began and extrapolating from that that is evidence of efficacy of their booster campaign. And yeah. So when you look to see who's getting infected in Israel, what they're seeing here and what they're showing here is that there was a climb in all age groups. But when they draw the line here of when the vaccines, the third dose boosters started in the older patients, then you see here that that group dropped in rate in terms of daily cases, and that the other groups, the younger groups, continued to climb after that. Okay. They also make the argument that, in fact, when you parse it out and say severe cases, that the severe cases also dropped. And again, this is the vaccinated with the 3 doses, and this is the unvaccinated. So they are arguing, and again, some of this, it's a little unclear how much noise and how much is going on. The other thing to point out here, and they didn't make an issue of this, but it's worth pointing out here, that Delta affected Israel very hard and fast early on, and that in fact that the rates were already dropping by the time some of this was happening, perhaps independently of this vaccination campaign, or perhaps because of the vaccination campaign. It's correlation, not clear whether there's causation. One other thing to point out is that their definition of severe case that they used was included anyone with a respiratory rate of 30 and above, or anyone with an ambient air, room air saturation of less than 94%. So their severe case definition was anyone, so if someone was having 92%, they counted as a severe case. And in fact, that they had many cases, and they didn't actually separate it out here. They had many cases of ambulatory outpatients who were classified as severe cases by this definition in Israel. So this is the Pfizer data, Pfizer slide here, but this is their summary, Pfizer's summary of what the Israeli data shows. And they said, if you compare it to 2 doses only, they say that you are seeing an 11-fold decrease in any infection and a 15-fold decrease in severe infection. And then back to the Israeli presentation, they said, based on this, based on modeling data, they said that they were predicting that if there's no boosters given, that this is what would have been seen in terms of active severe cases. And this is what they modeled with the initiation of boosters. And the dark line here is what they actually saw. Okay, I'm gonna stop there and take additional questions and comments about this. Before I do, let me take a look here. Within the US, it isn't a supply issue that is constraining things. So it's not someone's first shot versus someone else's 3rd shot. Matt, can you expand on what you meant by that? I think I understand, but I want to make sure I understand.
It was back when we were talking about like, is it better to have 50% of the community with 3 doses or like 70% of the community with 2 doses? That part of the conversation, I was just saying that this isn't a vaccine supply constraint. It's a number of people who are willing to get the doses. So within the United States specifically, not globally, but just within the United States, it's not a supply constraint. So it's not your 3rd dose versus somebody else's 1st dose.
No, totally agree. And this is an important consideration and conversation, because I think those that are in favor of it will say that we don't. It's not like giving the 3rd dose is taking. I'm saying the same thing, that we're taking away from someone's 1st dose. It's an acceptance issue, not a supply issue. Totally agree. Is there consensus regarding, Andrew's question, is there consensus regarding whether it is biologically plausible for SARS-CoV-2 to mutate in a way that would be able to evade vaccine-induced immunity? You answered your own question. No one really knows yet. But what they are, that is the worry. The worry is that eventually one of these will escape the vaccines entirely. Thus far, there is exactly 0 variants of concern that have been identified that completely evade or that significantly evade our most used vaccines. That's not to say that it won't happen. There's a lot of attention for a while on the Mu variant. There was the crazy worry about this Mu variant. The Mu variant has more mutations than Delta, and so people extrapolated and said maybe that means it's worse. Delta actually has fewer mutations than the Alpha variant, the B.1.1.7 one that was for a while the most prevalent one. So it's not correlated directly with the number of mutations. But you're totally right that it is a worry that eventually we'll get to one that doesn't. I feel better that the drug companies have already started tests and are producing trials of Delta-specific boosters, because that may become necessary, and because of the mRNA technique, they can do this relatively quickly. Annie, did Israel present data on deaths? They did not in their presentation. And I'm not aware of changes in death rates. The question, I believe, came up in the conversation, but I don't think that they presented data on it. And again, some of this, if you look at this, remember, deaths lag in terms of the data presented. And so it may be just too new, or it may get at this issue about whether or not we're measuring the same things. Other reactions to the Israeli data? What do we know about the incidence of rare side effects such as pericarditis after third vaccine dose? Unclear. According to the Israeli data, they said that they saw, I think they said they either saw very few or none at all is what they saw. But remember, this is confounded by the way in which they're doing this. They haven't vaccinated huge numbers of 30-year-olds yet. In fact, if you look at the Israeli data, the lowest rates of vaccine uptake, just as it is in the US, are among the 20 and 30-year-olds. So they do very, very well in the older groups, but like the US, their rates of uptake, even though better than ours, are not that great for the young. So we don't actually know the answer to that question, but great question. Other reactions to this? This, I'll tell you, these are the data that the administration was reacting to in August. I don't want to reveal my sources, but there's someone that I know is very, very plugged into the administration and shared some of these slides with me right before they made their announcement saying that the administration was worried about these data. So again, I'm curious about your reactions to these data, because this is what generated probably the big push to push for boosters.
I guess this might be kind of an obvious question, but now we know that there's no supply chain issue in providing enough vaccine. So would the arguments against giving a booster just be that we don't have enough evidence to suggest that it's required, and that like the question of what the threshold is in terms of like, is that kind of like, it's like more of a philosophical question of what the threshold should be to say we should do this? Like, because I feel like in medicine, usually if there's potential benefit to something and a low risk of harm, we usually do it, like in terms of other things. So I don't know.
Yeah.
Maybe
don't
cost,
get me started about some of
maybe cost of
yeah,
a booster
cost.
rollout if it didn't actually need it.
If there are any secret cardiologists on this group, I apologize, but you know, some of the SGLT2 data on some like HFpEF, I mean, really? Anyways, yeah, we do a lot of things without as much data behind it, for sure. But Arita, I want to come back to your question when we go back to the poll, because we're going to poll again in a little bit, because you're actually asking something super important, and I don't want to ignore it, but it'll be more obvious when we actually get to the poll. Other reactions? Sorry, did I insult anyone who wants to be a cardiologist? Mac, always with the great chat comment. Anyone? I'm curious. Just I'm not going to judge you one way or another. But is anyone convinced of the need for boosters based on this? Just yeah, I see some nodding. Anyone who maybe had been on the fence who now is convinced based on these data? If you are, I'd like you to unmute and just say what, maybe why. No idea what that model is based on. The model that's on the screen right now. Yeah. Modeling studies. If you follow the stuff, IMHE, like in our own backyard, tons of modeling. There's a well-known and well-worn phrase in the stuff, which is that all models lie, but some of them are still useful. IMHE got a ton of criticisms based on the early models because they were sort of all over the map early on, but they were the most effective models we had at the time. Like all models, like many models, they've gotten progressively more accurate in their modeling. And in fact, they were the ones that predicted back in May that we were going to have an August surge. And I remember looking at that thinking they were crazy, but here we are. So I don't know how the Israeli model is derived. Okay. All right. I realize I'm running out of time here. Okay, so let me quickly whip through the CDC presentation here on the vaccine efficacy. And so the CDC then presented at last week's meeting, and they shared what they're seeing in terms of the US. And what they're seeing is that, in fact, generally speaking, it looks like the vaccine remains effective over time against hospitalization. Over infection, it's dropping. It's very clearly dropping. The signal is very strong that the efficacy against infection is dropping in adults. But they overlaid an important caveat to all this, which is this in fact a Delta effect, right? And by mid-July, 94% of the circulating variants were Delta. Or is it an effect of the vaccines themselves? Are the vaccines waning in efficacy and that was always going to be the case, or is it Delta? If you look at pre and post Delta time points, the vaccine efficacy against infection has dropped all the way down to 39% in some studies, in some series. Against the hospitalization, it still remains pretty good, 75 to 95%. It still remains very effective against hospitalization. And this is another graph that shows that same thing. So even with dropping effectiveness against infection, it is effective against hospitalization. All right. So I have now presented you the exact data that they had. All right. And here is the question. This was the question that they had to vote on. And this is exactly how it was phrased. So same question you saw before. Do the safety and effectiveness data support approval of a booster dose administered at least 6 months after completion? Before you vote, if you haven't voted yet, let me point out that the Pfizer vaccine was approved as full approval. And so this was meant, again, as an expansion of their full approval when they were voting on this. So remember those criteria is that you have to have significant demonstration of efficacy in order to vote for approval. Sorry, I'm tilting the field here, I think, but go ahead and vote.
Just gonna give one more minute. Okay.
What do we see here? Yeah. Still, this was not that far off, I think, from our initial numbers. Okay. I'll tell you how if you didn't already follow the story, the commission voted, the advisory group voted 16-2 against approval. And the reason they voted, well, among the reasons they voted against approval was that they specifically called out that there was not enough data in the younger groups, that all the data were really focused around the older groups. So they regrouped, rewrote the question, and then they asked the second question here, which is, based on the totality of scientific evidence available. So again, they changed the phrasing here. Now they're using emergency use authorization, not the full approval language. Do the known and potential benefits outweigh the known and potential risks for a booster dose in those older than 65 or those at high risk for severe COVID? And then they later clarified to include healthcare workers or individuals at high risk for occupational exposure. So go ahead and vote on that.
Okay, we've got a quorum.
Okay. Yeah, pretty similarly, they unanimously voted yes on this one. They completely voted yes on this one. So they voted overwhelmingly no against the full authorization for boosters in all people over the age of 16. But when they constrained it more to older and high risk of infection and high risk of occupational exposure, including all of us, then they voted unanimously in favor of it. And so the wording, so I told you that ACIP has to approve of everything. So ACIP met yesterday, and if you watch the transcripts and if you watch the discussion, they were all kind of shrugging because as of yesterday, they didn't have a drug that was approved yet, or they didn't have anything to vote on, because FDA announced this final decision last night after the group met. And the group was discussing, they said, we don't have anything to vote on. We don't have anything to vote on. We don't actually have anything that's passed on to us. So they were trying to figure out what they would eventually have to vote on today. So last night, FDA took that language and said, yes, single dose for those older than 65, those that are at high risk, or those whose frequent institutional occupational exposure puts them at high risk of serious complications. The specific examples that were used in that conversation were healthcare workers, first providers, frontline providers. They also included teachers in the conversation. They also included long-term care facilities. So those are the kind of things that were meant when they were talking about institutional or occupational exposure. So literally, they may have already voted on it. They were meeting starting at 9 a.m. to vote on this issue. And so, I don't know, when we log off, we might discover that, in fact, it was approved. All right. I'm going to stop there and ask if there are final questions. But again, I think it's remarkable that after this whole process, that no one was coming up with anything that the group wasn't coming up with at last week's meeting, that these exact same issues, these exact same questions were the exact same things that they were discussing. So well done, all of you. You were asking all the right questions and had the right thought processes. I know we're about 2 minutes over. Any questions or reactions or thoughts?
I guess I just had a quick question. In terms of like the age 65, is it just because we know that that group, like in terms of the one graph I saw in this, I might have missed something, but like the rates of waning immunity are the same. We didn't have the earlier group in that one graph. It was like the same in all the age groups. So like, was this just kind of an arbitrary, like how, I mean,
yeah,
what they used to do that?
Yeah, so we didn't get to dig into it fully, but I think that there was really strong concern about the confounding by time, right? That because Israel was doing it in the older people first, that the majority of the data, so another table that I didn't show you, but there was some very contradictory data that was coming out that said, when you looked by subgroup, these things were there. When you looked overall, these directions of movement were opposite. And what it came down to is because it's confounded by this majority being in the older group driving the data. And so there's a lot of confounding and concerns about the confounding. And again, because of the higher level of data approval necessary for a full use authorization, they argued that they could not say that it met that bar, that high bar. And that's why they changed it. So again, the boosters that we're talking about are emergency use. They're not under considered full authorization. And the other thing to point out here is that they have not discussed Moderna or Johnson & Johnson in this. Moderna, just so you're aware, in their application, I mentioned to you that their dose was 100 micrograms versus Pfizer's 30. Moderna's application specifically asks to do 50 micrograms, and FDA turned them back and said, you need to provide us more data. Now, biologically, I cannot explain why they would expect a dose of 50 micrograms to be any worse than Pfizer's dose of 30 micrograms, but they required that they show more evidence. So they pushed them back and said, we need more evidence. Come back to us when you have more evidence. So again, kind of crazy in my mind, but that's the way the regulatory process works. Other questions, comments? I hope this was helpful and useful. This is, again, I think kind of fun to imagine yourself in the role of making those decisions.
Awesome. Well, thanks, John. I thought that was really great. I certainly learned a lot.
Cool.
Cool. Well, I hope everyone has a good rest of the morning. And of course, you know how to reach out to us if you have any questions or concerns.
Awesome. Maybe we'll all go and log off and see what happened.